from one generation to another without undergoing any alteration. The law can be illustrated with this example. When you have a tall breeding plant, a plant that is tall, then you cross it with a plant that is short or a dwarfish plant. When you cross them, you discover that the offspring that will be produced, which is the first filial generation, that's the first set of offspring that will be produced by that cross of a tall and a dwarf plant, we show that all the plants are tall, all the offspring will be tall. But on a further crossing, Mendel carried out this experiment. So on a further crossing of those tall offspring now, which will now result in a second generation of offspring, which is called the second filial generation. It shows that 3 of them are tall and 1 is dwarfish. That is, 3 are tall, then you have that dwarfness that was inherent in the parents, in the original parents, now showing in this second filial generation offspring. Now, look at this illustration. TT represents genes of the tall breeding plant, then the small TT represents the genes of the dwarfish breeding plant. Then the parent, so TT will cross the second TT, then all the gametes now, you have capital T, then small t, then all the offspring of the first generation are all tall, even though their tallness is not like this. The tallness of the TT, they have the capital and the small t. That's what you have in all the offspring. So it shows that it's a blend of what you have in the tall plant and in the dwarf plant, but they manifest all as tall plants. Now, when the tall plants, the offspring now self-fertilize, it's the capital T and small t, capital T and small t crossing, you discover that we have the 3 tall plants. 3 plants are tall. You can see them that they are tall. Then you have the dwarf plant. The dwarf that you have with the parents up here, you see have it here. And the phenotypic ratio, that is the physical appearance of the second filial generation, you have 3 of them that are tall and 1 of them that is dwarfish. And the genotypic ratio, now when you consider their gene, looking at the gene, you see that there is one that is actually tall, like the first parent here, that has the 2 double capital T. Then you have 2 of them having the T of the first filial generation, that is a mixture of the tall males and the dwarfness gene. That's what Mendel is talking about when he says they appear independently. That's the law of segregation. The tallness that overshadowed the dwarfness in first filial generation is said to be dominant. You know, that capital T is said to be dominant, while the dwarfness is said to be recessive. So the conflicting pairs of characteristics are called allelomorphic pairs. So the TT, capital T, and the small t being crossed. Now to the second law of Mendel, this Mendel's second law, which is also called the law of independent assortment of genes. The law of independent assortment of genes. It states that during gamete formation, the way one pair of alleles for a given character distribute themselves. So the alleles on different chromosomes are sorted independently during meiosis. During gamete formation, the way one pair of alleles for a given character distribute themselves does it as a separate unit and is inherited independently. So the alleles on different chromosomes assort themselves independently during meiosis. So they don't do it together, even though they are a pair, but they assort themselves independently as a separate unit. Methods of improving crop productivity. The methods of improving crop productivity include: first, you practice a good soil tillage system. Then you plant at the right time for your crop productivity to be improved. Then you adopt good planting methods like practicing in nursery, that is, you first raise your plants in the nursery, their seeds. Then when they've germinated well, then you transplant them to the field. regular weeding. So those are good planting methods that should be adopted for your crop productivity to be improved. Then effective methods of controlling diseases and pests. For instance, you use herbicide, you use pesticide, or sometimes you even plant early so that you can harvest early before the invasion of the pests. So those are the ways to improve your crop productivity. The next is improvement through introduction. Like I've explained crop improvement earlier, you can improve your crop through introduction, selection, and hybridization or breeding, which should be integrated into other cultural practices. Now, we move on to talk about animal improvement. Animal improvement is a process of transferring an inherited superiority from one domesticated animal to another of the same species. When you transfer an inherited character from one domesticated animal, or a character that is superior, a character that is superior in a domesticated animal to another of the same species, it is called animal improvement or animal breeding. Now, the aims of animal improvement are to produce animals with resistance to pests and diseases, then high feed convertibility, that is, they are able to convert their feed on a high rate, as the feed they eat, they are able to convert it to flesh, so it's better for the farmer. Unlike when they eat much, they have little flesh. So when they eat, the little they eat, they can convert a reasonable portion of it to flesh, so it's showing on their body. So you can be happy when you see your animal and say, oh, they are feeding well and they are doing well. Then the next aim is that they have good qualities. The animals have good qualities such as meat, then their skin, milk that you get from them, their bone, their hair, the heads. They have good. The animals are with such good qualities. Then such animals have high growth rate, then high yield. They reproduce fast. Then generally they have an overall improved performance. The methods of animal improvement, there are also 3 methods, and they are very similar to what we discussed in crop improvement. Introduction, selection, and breeding are the 3 methods of animal improvement, just as we have in plants. Introduction is a physical transfer of animals from their country of origin to another country. So when you bring a goat from Cameroon to Nigeria, that's introduction. So such animals that are transferred now are called exotic breed because they are not indigenous to this place. So we call them exotic breeds, while the ones that are in the country are called indigenous or local breeds. The breeds that are being brought in must have qualities which outweighs the ones that are indigenous to the country. So it will not make sense if you are bringing a crop or you are bringing an animal that is not better than what you have here, then there is no point wasting your money. So the animals you are importing or that you are introducing into the country must have qualities that outweigh what you have already in the country previously. Then there is an international regulation which stipulates that introduced animals must pass quarantine tests. So you don't just bring in an animal from another country and you just introduce this into the country. No, you take it through the quarantine and it must pass the quarantine test. So all introduced animals must be quarantined. So they will be checked in the quarantine process. Okay, what disease they have? Are they disease free? What pests are they carrying? What this, what that? So that we'll be sure that the animals are safe to live in the new country. Now, the period of incubation of such animals, that is in the quarantine, is dependent on the incubation period of the disease under consideration. So how long an animal stays in the quarantine determines the incubation period, that is the period whereby the disease that they are checking for takes to showcase or to surface. So that is the number of period that the animal will stay in the quarantine, so that they will be sure that even after that period of incubation has passed, the incubation period has passed and such disease is not noticed, then the animal has passed the test, then it can now be introduced into the country. Now, the advantages of introduction. First, it brings about breeds of animals with better qualities and they are now produced locally. So it's not that we want to eat a particular type of pig or want to eat a particular type of cow and we are now importing the cow. We can also produce such cows because of introduction into the new country. Then animals which were originally alien could be reared in a country. That is, animals that are originally foreign, they are not indigenous to this country, but they are strange. But now they can now be raised or reared in the country now. Then another advantage is that diseases and pests can be controlled since the animals must pass the quarantine test. So there's a level of safety or assurance that you have that diseases and pests, you are not bringing them in because the animals will have to go through quarantine and it must pass the quarantine test before it is introduced into the country. Then animals newly introduced will produce more than the local breeds, because before you bring in the animal, you must have been sure that it's a better breed before you introduce it into the country. Then we have breeds that have been introduced into Nigeria in the past. Breeds of cattle include the Fresian, the Holstein, Jersey, Aberdeen Angus, the Red Devon, Shorthorns, and so on. Those are the breeds of cattle that have been introduced into Nigeria. Then we have breeds of